Welcome everyone to the STEM Talk session number 4 of the 7th Polygens Symposium of Rising Scholars. My name is Yishim Merkan and I will be moderating today's session. The goal of the symposium is to celebrate and showcase the hard work of our Polygen scholars, all of whom have tirelessly worked on their projects over the past couple of months. We have 5 students presenting on a variety of topics today. A fellow member of the Polygens team will be scoring each student's presentation, which will help us determine prize winners after the event. Please type your questions in the Q&A if you have any questions for each speakers. We will have time at the end of the session for the speakers to answer your questions. Today's speakers are Mohna Chandra, Akhil Kalva, who is from Chatachuri High School and he's working on Alzheimer's disease. Sahvir Bhaskaruni, he's from the Academy of Science and he's working on virus infections. We have Taban Malihi from Newton High School from Massachusetts. She's working on polycystic syndrome, PCOS, and we have Neha Tarimadugu. Okay, Mohna, you're our first speaker. You can start.
Yeah, I actually wanted to ask about that. So there's like a slight problem with my presentation. I'm not able to back it up. So is it possible for me to go second?
Yes, of course.
Okay, thank you so much.
Okay. May I start now?
We are still waiting for Mohna.
Yeah, I asked to go second.
Okay. Now it's your turn, Akhil.
Okay. Hello, everyone. My name is Akhil Kalva, and I perform research on Alzheimer's disease in South Asians. I meant to do this project was with Jared Hinkle. Here's just a little bit about myself. I reside in Johns Creek, Georgia, a small suburb in metropolitan Atlanta. I was born and raised in California, and I moved here when I was about 2 or 3 years of age. I attend Chattahoochee High School, and I'm currently a junior here. A lot of the classes I take in school are tailored towards STEM, and I wanted to pursue my interests in this field outside of my school environment, so I wanted to perform some research. I have a personal connection to Alzheimer's as I have seen and observed people with AD and seen how their lives have changed due to this condition. I wanted to focus my research specifically on the ethnic group I am part of, which is the South Asian community, as I know they're underrepresented when it comes to focus on research. So here's the agenda for the presentation. I'll first introduce the issue, discuss the different genetic risk factors such as APP, the amyloid beta proteins, APOE, and tau. Then I will discuss the different lifestyle factors that are more controllable, such as exercise, diet, and the heart. Then I'll put all this research into perspective and provide future directions and steps to move forward towards. Alzheimer's disease is the most common type of dementia. Dementia is a general term for loss of memory, language, problem solving, and other thinking abilities that are severe enough to interfere with our daily life. Alzheimer's specifically impacts memory the most. It is caused by the accumulation of plaque in the medial temporal lobe. The medial temporal lobe is inside the middle of the brain and has a lot of distinct functions. The hippocampus is a part of the medial temporal lobe and is responsible for memory. These plaques impact the hippocampus the most by blocking its ability to send and receive signals. We'll go into more detail about the specifics of these plaques later in the presentation. Alzheimer's impacts specific demographics the most. Age is the largest factor, as people over the age of 65 are more likely to develop Alzheimer's. Women are also more likely to develop AD compared to men. The picture on the left displays a very brief photo of the difference between a healthy brain and Alzheimer's disease brain. As those little orange circles on the right side represent the plaque, which can disable the ability of these neurons to be able to relay information as quickly as they need to. On the right hand, there's a chart that displays the common Alzheimer's symptoms, and some of these include confusion, difficulty solving problems, poor judgment, and most importantly, memory loss. All of these can hinder the ability of a person to continue with their daily life tasks. I will now discuss the genetic risk factors. Amyloid beta peptides, or AB for short, are especially important in the role of neural growth. They can help repair damaged neurons and protect the body from infections. However, it is deemed as one of the largest causes of AD. It is developed in the AP protein, or the amyloid precursor protein, which we'll discuss later. It can form in plaques, and this can disrupt cell function. These clumps can disable the ability of different neurons to be able to send information as they block the pathway for the neural signals to send. They're caused by a variety of factors, and the overproduction of them can lead to some profoundly serious mental conditions. They are found in the 36 to 43 amino acids that are processed by the amyloid precursor protein. Next up is the amyloid precursor protein. It's a transmembrane protein that plays a major role in the regulation of several important cellular functions, especially in the neural system. However, specific mutations can cause a proteolytic cleavage. A proteolytic cleavage is the process of breaking the peptide bonds between specific amino acids. This can result in the protein being too long or being more active than it needs to be. These mutations can also release excess levels of AB peptides, which once again lead to plaque buildup and then in turn lead to neural degeneration. The diagram on the left here displays how these different AB plaques form from AP. Different inhibitors, such as the BACEI inhibitor and the gamma secretase inhibitor, can cause AB monomers, and a lot of these monomers can stack up to form oligomers. These oligomers can eventually form into plaques. The chart on the right displays how exactly the AB peptides can lead to Alzheimer's. The increased production of these AB peptides are caused by the APP mutations. This can lead to the aggregation of these peptides, which increase the likelihood of phosphorylation of tau, which we'll cover in the next slide. These plaques can form the tangles of tau, which lead to cell death and eventually Alzheimer's. Now on to the impacts of Alzheimer's regarding tau. Tau is a microtubule binding protein, and its main function is to stabilize neurons so they don't overfire or damage themselves. The largest issue when it comes to tau is the hyperphosphorylation. Phosphorylation is necessary for a body to function, as its attachment of a phosphate group to a molecular ion is required for a body to process energy, such as ATP. However, too much of this process is called hyperphosphorylation. Hyperphosphorylation can lead to the dissociation of tau and other neurons. This dissociation can lead to neural degeneration, memory loss, and learning disabilities. All of these are very common effects of Alzheimer's. The image on the left displays the different stabilities of tau in a healthy microtubule compared to an Alzheimer's microtubule. In the top one, the healthy microtubule, you can see that the tau helps stabilize the protein and keeps it intact. However, in the Alzheimer's one, the tau tangles up on itself, allowing for the microtubule to spread in many ways, making it difficult for it to perform its function. The diagram on the right shows the brain, the different steps from going to a healthy brain to one that's impacted by tauopathy. It starts with tau degeneration, and with added oxidative stress, it can cause disulfide bonds in tau to break, which leads to tau aggregation and the loss of neurons, which is the cause of Alzheimer's. Now to the impact of APOE, or apopolipoprotein, on Alzheimer's. APOE is a protein that is a major cholesterol carrier, meaning it is responsible for distributing cholesterol to the different parts of our body. It also supports lipid transport, which is important in maintaining body temperature and ensuring our muscles are held together. However, a specific allele called APOE4 increases the likelihood of an Alzheimer's disease prognosis. The E4 allele is present in approximately 15 to 25% of the general population, according to Mayo Clinic. Having this allele increases the risk of Alzheimer's disease 2 to 3-fold. On top of that, having 2 or 3 of these alleles increases your risk by approximately 8 to 12-fold. This means that it's not a guarantee, but it's a much higher risk of being of being diagnosed with Alzheimer's. The allele can cause a lot of the risk factors we've discussed above, such as tau degeneration, the buildup of AB plaques, and increased metabolism of APP. The image on the left displays an astrocyte. When the APOE3 allele is present, it shows that it is sufficiently transporting lipids to the neurons, keeping them healthy. However, when an APOE4 allele is present, or an aging one, it supports an inefficient amount of lipid transport, and the cell goes through neural degeneration. The diagram on the right displays exactly how the APOE4 allele can cause all the things that we've discussed. It can lead to tau degeneration, which leads to AD. It can cause the buildup of AB plaques, which can lead to AD. It can lead to altered APP metabolism, which can lead to plaque buildup, which in turn leads to AD. Overall, the APOE4 allele can cause all the issues we've discussed above. Now moving on to a more controllable and lifestyle factors of AD in South Asians. Heart risk is a large factor in the controllable aspects of Alzheimer's disease. Low-density lipoprotein is one of the 5 major groups of lipoproteins that transport all fat molecules across the body. It is proven that having elevated levels of this can increase the likelihood of AD. Some of the most common heart diseases that are more prevalent in South Asians include high blood pressure, atherosclerosis, and high blood cholesterol. It is important to understand this, as South Asians have the highest risk per population of heart disease. It's commonly known that the heart and the brain do evolve together and impact each other, vice versa. Diet also has a link to AD. The foods South Asians eat is notorious for the carbs and fat that they have. This carbs and fat lead to fatty deposits, which can decrease blood flow to the brain, which can lead to neural degeneration. Diabetes also has a high correlation. It's one of the most common conditions to South Asians. The inflammation diabetes has causes the lack of blood flow to support the blood vessels in the brain, which can cause neural degeneration, which leads to Alzheimer's. Activity and exercise is also important. Physical activity can reduce the risk of AD. People who exercise more have a higher spatial memory, larger hippocampus, less cortical decay, and improved attention speed. So the conclusions I drew is that age, genetics, and lifestyle play a large role in the diagnosis. Genetic risk factors and lifestyle factors are equally as important, and lifestyle factors are ones you can control. Some future directions I have is that we could decrease the likelihood of Alzheimer's in South Asians as more research needs to occur on South Asians specifically. Few papers and publications are focused on this area plagued by cardiovascular disease. As established, cardiovascular disease relates to AD, so focusing on that perspective on the prognosis of AD can be effective and decrease its likelihood in South Asia. Overall, equal intervention is needed because our ethnic group, the South Asian community, does not get equal opportunities to be healthy and to be cared for, so they're at a higher risk of Alzheimer's disease. Anyone have any questions?
Thank you so much, Akhil. It was a great presentation. We will get to all questions at the end of our session.
Okay.
And our second speaker will be Sahvir. Are you ready, Sahvir?
Yes, 1 second. Let me get my
Excellent.
screen shared and I will be presenting.
Okay.
All right, can everybody see my screen?
Yes.
All right. Hi, everyone. I'm Sai Sahvir Bhaskaruni, and my project is designing a small molecule inhibitor of the PCDH1 protein in order to combat Andes orthohantavirus infection. So to start off, let me introduce what the Andes orthohantavirus is. The Andes orthohantavirus is a negative sense RNA New World hantavirus that's endemic to Argentina and Chile, but present in most of the American region. Andes orthohantavirus causes hantavirus cardiopulmonary syndrome, which has a 36% fatality rate. And it is for this reason that the WHO has recognized it as an emerging public health threat, meaning that we have to make moves to address it now before it becomes a true public health threat. Andes orthohantavirus is transmitted by rats to humans through rat excrement particles stirred into the air or direct contact, most commonly in areas which are highly crowded, polluted, have little sanitation, and less prevalence of medical professionals. There's also currently no FDA-approved treatment or vaccine to combat Andes orthohantavirus infection, meaning that we need to start moving quickly to develop treatments and vaccines to prevent this infection. So before I start getting into my procedure, there is some background you need to know about how the Andes orthohantavirus binds to our human cells and really infects our bodies. The Andes orthohantavirus has two glycoproteins, the GC and the GN glycoprotein, but we're going to be focusing on the GN glycoprotein. This glycoprotein is on the viral cell surface and is responsible for binding, infusing to our cells, specifically our endothelial cells, to the protocoadherin 1 surface level protein. From here, it fuses to this protein, and then that starts the process of viral cell fusion, which starts the entire process of infection. So if we can prevent this step, then we can prevent cell infection. So now this works by the N terminus, so the beginning of this glycoprotein, which starts right here. This area, these peptides here bond in this region. So this is protocoadherin 1, the surface protein on our cells, specifically ectodomains 1 and 2 in this red box here. And what this surface level protein does, it binds to here, then fuses the virus to this protein, causing and allowing the virus to enter into our cells. So if we stop this process, develop a competitive inhibitor, so something that can block this from occurring, then we can theoretically prevent hantavirus infection. Now, in order to do this, I first did a reference docking of the amino acid sequences TIY and TI. So this is that N terminus. These are the regions, that small subsect of the protein, the GN glycoprotein, that's responsible for this fusion. And this is the threonine isoleucine tyrosine compound and the threonine and isoleucine compound. Then I did a diagnostic docking where I added oxygens and nitrogens and studied those effects to determine where the additions were needed to make it a more effective competitive inhibitor of the viral cell membrane fusion. And then I did some experimental docking where I designed compounds that adhere to Lipinski's rule of 5 for druggable compounds and tested them and compared them against what I determined in the reference docking to see if we could possibly create a competitive inhibitor of the GN glycoprotein to prevent Andes orthohantavirus infection. This study aims to be a proof of concept study and to show that it's possible to create a competitive inhibitor and to help further the development of a true drug candidate that we can all use to prevent and treat Andes orthohantavirus infection. Now, I first needed to decide whether to use TIY or TI as a drug backbone. So I did a few docking studies where I docked TIY and TI to that PCDH1. If you remember 2 slides ago, that in that red box on the right side of the slide, in the between the ectodomains 1 and 2. And then what I found was that TIY was more effective. Now, I did this by looking at the scores. So now what you see here is the scores. These are negative numbers. These negative numbers indicate the binding affinity. So the more negative the score, the better it binds. A good analogy to this is it's like a sticker. The more negative the number, the stickier the sticker is. And we wanted to have a really sticky sticker to prevent that GN glycoprotein from attaching to our PCDH1 proteins on our cell surfaces to prevent Andes orthohantavirus infection. So I concluded that the TIY drug backbone would be the most effective, and then I started modification from there. The next step was the oxygen versus nitrogen diagnostic docking. So now what this is here, what this is I added oxygens to each of the ends here that didn't already have an oxygen or a nitrogen linked to it onto the free hydrogens, and then tested and cataloged to see where H bonds form. So where did the bonds between the PCDH1 protein and this compound here form, and where can I mimic it? And which of these additions were the most beneficial, so that I could use it in a potential candidate in the future sort of design and analyze and understand what I needed to add to this compound where. And then I repeated the process for nitrogen. I included the binding scores here just to get a picture of the fact that oxygen was in fact a little bit more effective than nitrogen in eliciting a stronger binding response. Those scores are similar. And you'll see by the next slide how I was able to come to the determination that the additions of oxygens are more beneficial. So moving on to the next slide, this is where I really started the legwork of my project with designing potential small molecule candidates to inhibit the PCDH1 GN glycoprotein binding. So I conducted about 30-ish trials and 30-ish designs where I took the data from the previous diagnostic dockings studies and applied it to about 30 trials, which adhere to the rule of Lipinski's rule of 5, and then just to see whether I could come up with a small molecule that was more effective at binding to this region, so had a lower binding or delta G value to see if it would be more effective. And what I found was that I was successful in 11 of the attempts, and you can see these here. And I've bolded the ones that are significantly above or significantly below and have significantly better binding affinity. And I think this picture is great. It's a great demonstration of what I'm really trying to do here. So this is trial 10. So this is a delta G value of -6.4. And this is just some modifications that I made. So you can see this here, 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 and here. So the oxygen modifications that I added. And what it does is it binds and it docks to this particular region. This is the EC1, EC2 region, this particular docket where the GN glycoprotein actually fuses. And because it's taking this space up here, right, it's preventing the GN, it will prevent the GN glycoprotein from binding in this region, potentially and probably protecting us from further Andes orthohantavirus infection. And this can then be confirmed with in vitro and in vivo studies to test that, its actual efficacy. And it's these 11 candidates here that they can really be beneficial in guiding future development of a true candidate as a proof of concept, as I mentioned earlier. And that's kind of what I did. Thank you guys for listening, and please let me know if you have any questions. Thank you.
Thank you so much, Sahvir. It was a great presentation. Good job. And our third speaker is Mohna. Are you ready, Mohna?
No, actually my issue hasn't been solved. I'm not able to present it for some reason. So is it okay if I go after this?
Okay,
because I'll
okay.
switch to another device.
Okay, no worries.
I'm so sorry.
No worries. Taban, are you available? Are you ready?
Yep.
Excellent. Excellent.
Okay. Give me 1 second. Oh, sorry, my mom's in the background. All right, I'm going to just share this presentation and share my screen. Just a second. Just 1 second, sorry. Okie dokie. Here we go. All right, I'll start whenever we're ready. Is everyone good to go?
Yes, we are listening.
Excellent. So hello, everyone. Thank you for coming to my presentation. I'd like to thank my mentor, Katie, and my family who support and inspire my interests. My project is a comprehensive literature review of peer-reviewed medical journal articles analyzing the relationship between metabolic syndrome and PCOS. So the reason I'm focusing on this right now is that there's a global epidemic of metabolic syndrome, but even though incidence rates are rising, there's a real lack of awareness around the condition. Additionally, there are disproportionately high rates of insulin resistance amongst BIPOC and Hispanic communities. However, this is not really reflected in the literature, which is highly exclusionary of these communities. Moreover, you can see high rates of insulin resistance within the PCOS community. These lead us to believe that there is a higher risk of developing PCOS within communities that are susceptible to insulin resistance, which makes both the PCOS and metabolic epidemics relevant issues that the medical community needs to address. So what is PCOS? It is an endocrinopathy, as you can see. It's a hormonal condition. It affects approximately 2 to 20% of people's uteruses, and it results in these things on ovaries that are cysts. Essentially, they're very painful, small pustules that can explode at any given point, causing a lot of pain for patients. And globally, the impacts of polycystic ovarian syndrome are mainly infertility. This affects a very high rate of PCOS patients, up to 80%, as well as high rates of mental health disorders such as depression and anxiety. There's also insulin resistance over here, which is also known as metabolic syndrome. It's a precursor to type 1, type 2, and gestational diabetes, and it affects a very large percentage of people worldwide, from 15 to 46%. Okay, so here is the evolution of the diagnostic criteria for PCOS over the years. The Rotterdam criteria is the most widely used, despite being from 2003. However, the NIH criteria is the most recent. It adds upon Rotterdam by establishing 4 phenotypes, as you can see down here, that show different combinations of different symptoms of PCOS, because of course, not every manifestation of PCOS is going to be the same. As you can see, these symptoms include oligo or anovulation, which are irregular or lack of menstrual periods, hyperandrogenism, which is excessive presence of testosterone, which can often result in things like hirsutism, as you can see over here, or acanthosis nigricans, which is hyperpigmentation on the back of the neck, and as we saw on the previous slide, polycystic ovaries. However, there are systemic issues that create gaps in research and detection. Namely, the vast majority of studies don't incorporate phenotypes into their analysis, which is really harmful in situations where, for example, clinical populations frequently include phenotype A, in contrast to unselected populations, which frequently include phenotype C. The research really isn't representative of the population at large, as the literature suggests. And the root cause of this, according to one study, is a lack of methods and standards being standardized across the board of detecting the symptoms themselves of PCOS, for example, hyperandrogenism. That is why, for example, because these things are largely unreliable and diagnosis is subjective at this point, overall, this is really harmful to patient well-being because we can't detect things effectively. And additionally, the system itself puts a really heavy burden on patients. It takes 33% of patients over 2 years of consultation just to get a diagnosis, and almost half of patients have to consult 3 or more health professionals throughout the whole process. And as you can see, the journey to diagnosis, this presents a significant challenge to anyone who cannot afford the requisite immense amounts of time, money, and effort, which creates a really large socioeconomic barrier to accessing treatment. So what are the causes of PCOS? Right now, the pathogenesis is highly contended within the community, but the general consensus is that the hypothalamic pituitary gonadal gland is dysregulated in PCOS patients. Essentially, this happens because over here, the hypothalamus, it secretes GnRH, which stimulates the anterior pituitary gland to release hormones called LH and FSH. These stimulate the ovaries to release estrogen and other sex hormones, and all of this creates a feedback loop for the system. However, in PCOS patients, there's an imbalance when LH is elevated in proportion to FSH, which causes the symptoms of PCOS. Okay, what are the potential causes of this dysregulation, though? So there are conflicting hypotheses, but most sources agree the disordered feedback loop can be traced to the ovaries down here, where basically insulin acts as a gonadotropin and stimulates the ovarian theca cells. This accomplishes 3 things. First, it increases levels of testosterone. Second, it decreases the amount of estrogen your body is producing. And thirdly, it impairs the signaling of your ovaries, which disrupts the feedback loop. And it's really no surprise that insulin resistance is a major risk factor, therefore, for PCOS, because there are many reviews and studies that have found, for example, that 20 to 24% of girls with type 2 diabetes suffer from PCOS, which makes them twice as likely to have PCOS than those without diabetes. Evidently, this is a very very prevalently and closely linked relationship between the two diseases. And interestingly enough, PCOS itself can also perpetuate a cycle of insulin resistance. Not only does insulin resistance cause it, but when you have higher androgen levels from PCOS, this increases the levels of the free fatty acids in your bloodstream, and this causes your cells to perceive glucose deficiency, your body not having enough blood sugar, and that causes them to increase insulin production. This causes hyperinsulinemia, which is the result of insulin resistance, and then your body goes through this entire loop again. So it's a really it's a self-corrupting system in a sense. And as you can see here, there are other potential causes of HPG axis disturbance other than insulin resistance. For example, there's high levels of the protein kisspeptin, low levels of sex hormone binding globulin, which basically is a protein that it primarily regulates your circulating sex hormones, but it has also been associated with regulating your glucose levels and your blood sugar. And that's really important because if we can study like how low levels of SHBG, they're a causal factor for both PCOS and type 2 diabetes that shows a very close link between the two diseases on a new level. And additionally, there are genetic variants that have been detected, although research is limited for all of the causes on this slide. For example, there's loci 2p16.3, 2p21, and 9q33.3. All of these are associated with PCOS. And that's really important because when you look at type 1 diabetes, genetics plays a very large part in the development of that disease. And 24 to 33% of women with type 1 diabetes develop PCOS and PCOS-related traits. So that indicates a potential genetic relationship that really warrants further research. Additionally, there are in utero influences that children of PCOS patients can develop PCOS from if their parents have PCOS. So for example, there's 30% higher levels of 5-alpha reductase activity, which is an enzyme that increases testosterone production, which causes hyperandrogenism. As we know, that's one of the symptoms of PCOS. There's also high prenatal exposure to testosterone, as well as anti-Müllerian hormone. These both cause, in the children of PCOS patients, a higher chance of developing both insulin resistance and PCOS, which means that both of these are intergenerational issues. So what are the solutions for PCOS? So right now there's no cure, but the most common solutions are lifestyle changes in metformin, mainly because they're most affordable, and it helps most patients who have high levels of insulin resistance or obesity because, for example, exercising and maintaining a healthier diet really helps solve back for a lot of the weight issues, which then helps people maintain their insulin resistance issues. And as we saw before there, it really is a feedback loop. So if you can try to solve for one part of it, you can more or less help back solve for the overall issues that are compounding the system. But unfortunately, these are not long-term solutions, and these by no means really work for everybody. This is just the vast majority of people can benefit from these if they have obesity or insulin resistance. There are more expensive treatment options, for example, bariatric surgery, hormonal therapies, and laparoscopic ovarian drilling, but these are very expensive. There's varying rates of success. And right now, there's not enough data to concretely understand how effective they are. We really need more research to understand and develop, not only understand the current treatments that we have, but like develop new treatments and hopefully someday find a cure for this extremely prevalent disease. So this is a product of the literature review on a more analytical level. The discourse that I noted whilst in the process of research, I think really warrants calling out. For example, PCOS symptoms, including obesity, hyperandrogenism, all of these often result in presentations of an individual that run counter to normative societal expectations of feminine gender presentation, which means PCOS patients can really struggle to reconcile their experiences with society's expectations. And there's also social pressure related to having children, which really harms the mental health of oligo and anovulatory patients. And both of these sort of understandings of the self are related to medical institutions and professionals who are contributing to the literature discourse, who commonly assume, for example, that the primary concern of all PCOS patients is fertility, which is reductive and doesn't necessarily affect all patients and their approach to this very widespread disease, which means that this is a discourse issue that needs to be addressed at the root. Additionally, this one was really, really important to me to point out. There's a lot of cisnormativity and the exclusion of trans, intersex, and chromosome variant individuals, especially if they're BIPOC within medical discourse. For example, there's one patient that described their experience with PCOS as both physically and psychologically painful because the medical institution was not built in any way to address their personal experiences within it. And additionally, the intersection of transphobia, intersex phobia, and PCOS-related hyperandrogenism, these all result in, for example, literature still operating primarily within biological sex binaries, which is very exclusive towards intersex individuals. And one study even citing hyperandrogenism as an unfair advantage for women in athletics, which is just citing transphobic and intersex phobic discourse and implications. A recent paper has outlined why that is incorrect and why in general, we just need to be more aware of how the literature affects our societal understandings of PCOS. So in a nutshell, my message to the scientific community is that your literature, it really matters and it has real world implications for patient care. So looking at PCOS and metabolic disease as a whole, the literature, of course, indicates a very close link to metabolic syndrome for PCOS, which indicates solving for one necessitates addressing the other. But we also have to look at, for example, the really ineffective diagnostic processes at the moment and treatment options, as well as the institutional racism, socioeconomic barriers, and transphobia, intersex phobia, which are direct results, and contribute to a feedback loop of exclusive literature and medical institution discourse. And we really need to address not only these, but the research gaps that compound these systemic inequities while discounting different phenotypes of PCOS. These often ultimately result in negative effects on patient well-being, and it really locks out the opportunities that we all have for innovative and specialized solution making for PCOS, which, as we know, is a very prevalent condition and will continue to become more prevalent as the metabolic epidemic worsens. Overall, this really comprehensive review indicates a need for broader and more intersectional lenses of analysis pertaining to PCOS. Thank you. And here, just scrolling through the references, because this is a literature review.
Thank you so much, Taban. It was awesome.
Thank
It was
you.
awesome.
All right.
Our next speaker is Mohna.
Yep, I'm ready.
Thank you.
Okay, I'll just share my screen. Yeah. Hi, I'm Mohna. First, I would like to apologize for the delay. I had a few issues with my laptop, but yeah, today I want to present my research on modern cancer treatments, integrating treatments and technology. So a little bit about myself. I am from India and I lived in California for like a bit as well. And this is something that's really important to me because I plan on being a physician in the future, specifically a pediatric oncologist. Yeah. So who is cancer? Cancer is a disease in which some of the body cells grow uncontrollably and spread through other parts of the body. Cancer is the second highest cause of death worldwide. And the paper that I've written with my mentor, Dr. Samuel Hart, highlights a few innovations in cancer treatments over the past few years that have helped improve the quality of lives of patients and increase mortality ratios. So what has cancer done to human lives over the years? Cancer drug discovery and treatments have undergone extraordinary changes. The understanding of cancer has advanced considerably and the treatments are less punishing. Modern cancer treatments are characterized by integrating advancing technology, drug discovery, and treatments. Over the years, the doctors and scientists have tried to modify and innovate on the existing cancer treatments based on past mistakes or gaps in already existing treatments such as surgery, radiation, and chemotherapy. Cancer research and treatments have taken years to be used with patients officially, and the future of oncology will require clinical trials and combining treatments to maximize their benefit of both into one therapy, such as oncolytic viral therapy and the CAR T-cell therapy, which I discuss in this paper. New and improved times during cancer treatments. So what are the new and improved times? like it is the transition from the treatments that we used in the past and how they were modified according to the patient's needs and result in limitations to create modern treatments. This section of the research paper focused on the new and improved times in cancer treatments that are pushing along existing lines of attack, opening entirely new lines of attack, and coordinating the treatments along multiple lines of attack, such as oncolytic viral therapy, personalized medicine. Throughout this paper, I've covered how cancer treatment has evolved in the past 50 years and highlighted the benefits and innovations and intersections of the selections of treatments. The modern cancer treatments that I've covered in this paper are oncolytic viral therapy, personalized medicine, CAR T cell therapy, and proton beam therapy. 10 million people. That's the number of people that have died of cancer just in this past year. And what can we do to change that? Oncolytic viruses. Oncolytic viruses are a form of immunotherapy that uses viruses to infect and destroy cancer cells. Oncolytic viruses offer a benefit for treatment because you can inject an individual tumor and you don't need to define a primary antigen that is present in the tumor. Immunotherapy is an evolving and promising cancer treatment, and it works by stimulating the immune system. Immunotherapy is a type of cancer treatment that helps you fight your immune system. The immune system helps your body fight infections and other diseases. It is made up of white blood cells, organs, and tissues of the lymph system. Oncolytic viral therapy also saves a lot of time as you don't need to define the primary antigen. So how does oncolytic viral therapy work? The treatments that can fuel the body's production of cancer-fighting cells or help the healthy cells identify and attack cancer cells. The oncolytic viruses will induce a very strong immune response, largely built on the immune system's response to the viral components, but there will be viruses spreading to the tumor antigens, and so it continues to be very exciting as an adjacent to immune therapy. In the early days of development of oncolytic viruses, the scientists were looking for that direct killing by the virus of the tumor cells, but now we understand that it is probably the immune response. So you can see rejection of uninjected lesions, which is an episcopal effect. So the healthy undamaged cell so from the tumor cell, it is converted to a healthy undamaged cell by the destruction of the tumor through oncolytic viruses. The oncolytic viruses destruct the tumor microenvironment and the systemic anti-tumor immune response, which doesn't let the tumor grow in that environment. Personalized medicine. Personalized medicine is a form of medicine that uses information about a person's own genes to prevent, diagnose, or treat a disease. In cancer, personalized medicine uses specific information about a person's tumor to help make a diagnosis, plan a treatment, and find out how well a treatment is working or make a prognosis. These have made us think and reflect on a difference, and as analysts plan more compelling medications, they've utilized hereditary data to create tests for cancer and ways to avoid it. The goal of personalized medicine is to use the right drug at the right dose with minimal or no toxicity for the right patient at the right time. A personalized treatment may influence healthy cells and cancerous cells too. Cancer, moreover, has been observed to occur in people who have no family history of cancer than people who do have family history of cancer. So why is personalized medicine even needed? So despite the fact that DNA from different cells is the same, genes that code for one organ and its cells behave differently from cells in other organs. Different cancer tumors may have the same DNA, but gene expression is different for different tumor types. Technologies such as gene expression microarray allow us to examine the gene expression profile of hundreds of genes at a time to distinguish a cancer-associated gene expression profile from a normal profiling. Personalized cancer therapeutics have less side effects than other sorts of treatment. This is often since it is planned to be more particular to the person's needs. But there are a few limitations of personalized medicine. It is widely known that the presence of more than one molecular change collectively with the tumor heterogeneity continually limits the use of monotherapy, and that can justify the death of advantage derived from precision medicine. Despite a promising organic historical past justifying precision remedy, positive poor effects have been highlighted obstacles with inside the molecularly pushed remedy strategy. So this is a clinical case that I researched on. There was a Shiva trial in India in which 741 cancer patients recognized with any form of strong tumor had been screened for molecular changes. In the changes remedy of desire, as opposed to focus on the remedy decided on in line with their molecular profile, should function as a paradigm for what continues to be incompletely understood while making use of the precision remedy in most cancer sufferers. But there was no advantage in median survival rate seen, and it became found in molecularly oriented sufferers as opposed to the traditional method. Nevertheless, this also helped us make an important evaluation that displayed the positive obstacles probably answerable for the failure of this method. Moreover, the factors of this method used in the trial are questionable, which includes hormonal remedy in closely handled sufferers or non-specific inhibitors while focusing on positive molecular changes. What is proton beam therapy? Proton beam therapy is a form of radiation treatment that's used to destroy tumor cells. Instead of using X-rays like regular radiation treatment, it uses protons to send beams of high energy that can target tumors more precisely than X-ray radiation. Proton beam therapy is often used in pediatric oncology as it reduces the risk of long-term brain damage in children and reduces the side effects of radiation usually tends to have. Children with malignancies of the central nervous system, brain and spinal cord, and the eye, such as retinoblastoma and orbital ribosome sarcoma, may get proton treatment. Over the past few years, pediatric oncology has improved dramatically, and 5-year overall survival is about 80%. In the United States alone, there are roughly 270,000 childhood cancer survivors. Although from personal experiences, I know that proton beam therapy is very expensive because I've shadowed multiple oncologists in the past few years. And in India, proton beam therapy is not very common. There's only one hospital where it's used, but since I did go for a medical internship to Vietnam, I did see proton beam therapy happening in pediatric patients, and the results were absolutely phenomenal from the basic chemotherapy or radiation therapy used, because there's obviously less chances of the tumor recurring and there's less consequences. So this is how it was done, essentially, and yeah. So the advantages and disadvantages of proton beam therapy. Advantages is that the radiation injury to the healthy tissues is less likely. Additionally, some forms of tumor may be able to receive a greater dose of radiation, which may prevent the tumor from growing or spreading. Long-term consequences associated with traditional radiation therapy may be reduced with fewer and less side effects, such as low blood count and fatigue. Disadvantages. Up to 2/3 of the patients will develop long-term toxicity, some of which will be fatal and are more likely to occur in pediatric cancer patients. Long-term consequences include growth abnormalities, second malignancies, neurologic complications, reduced IQ, cardiac and pulmonary toxicities, and infertility may occur, but it can all be used for the use of proton beam therapy. Combination therapy. I'll be talking about the combination of CAR T-cell therapy and oncolytic viral therapy. The combination therapy using oncolytic viral therapy and CAR T-cell therapy is effective because oncolytic cancer therapy has the ability to specifically target cells and replicate in them, which is unusual. One of nature's most destructive pathogens are viruses, and we are using that to target cancer cells rather than using it to cause a disease. CAR T-cell therapy involves re-engineering the immune system to see a cancer, and we are able to re-engineer T cells to be specific to any target that we're interested in. CAR T-cells are revolutionizing the field of cancer therapy, providing hope for a cure in patients with previous refractory cancers. But there are a few challenges of combination therapies. However, despite the stunning results of CAR T-cell therapy in patients with hematologic malignancies, this approach has shown little effect in patients with solid tumors. Recent clinical trials demonstrated that CAR T cells are able to infiltrate the tumor mass and extend antigen-directed cavity. The first example of this was a liquid cancer targeting a protein called CD19. There is no such thing as a CD19 for solid tumor because they're all heterogeneous. Another undertaking is the immunosuppressive tumor microenvironment, which may be the bodily boundaries which can provide stable tumors to save good enough T molecular infiltration. So the concept that we had to turn down the use of oncolytic viruses as a vehicle to supply a goal of interest. It acts like a car. The effects of CD19 on tumor cells. They have been picked out from chimeric viruses that re-engineered virus into specific CD19 with the concept that if it infects the tumor cells, mainly consisting of the CD19, it will supply the CD19 in order to provide a protein trip to the floor. It may be centered through the simplest vehicular T molecule that might not have been inflamed through the virus. It is the aggregate of each recruits that indigenizes the immune system to the tumor to target the cells that might not have been centered through the virus. The initiation of these scientific trials and the interesting part of the improvement method is that within the destiny, we can use this treatment on any stable tumor or any affected person. So this is a picture of how the CAR T cells therapy is usually works. So the CAR T genes are inserted into the T cell via the vector, and they're expressed due to the hinge and transmembrane region acting upon the antibody-derived recognition moiety, and then it forms the anti-CD19 CAR protein and the CAR expressing T cell. So there are a few advantages and disadvantages of each of the therapies that I did talk about. Oncolytic virus therapy aids in the tumor the ability to generate virons rapidly and genetically engineer additional genes that promote anti-tumor immunity, increase the tumor cell susceptibility to ionizing radiation. But there are some limitations, such as chills, fatigue, flu-like symptoms, injection site pain, nausea, and fever, relatively poor intranumal penetration by the oncolytic viruses. Personalized medicine is highly specific and reduction of adverse reactions, but there's a lack of information regarding the long-term side effects of personalized medicine. Proton beam therapy compared with the traditional radiation, protons have unique properties that allow doctors to get better targets of radiation to the size and shape of the tumor. The proton beam kills the tumor and spares more of the healthy tissue. But there is hair loss, fatigue, skin redness around the part of your body being treated. The combination therapy of oncolytic viral therapy and CAR T cell therapy increases the permeability of the tumor cells for T cells, reducing the disturbing effects of immune system, increasing the success chance in the treatment of hazardous disease. Sometimes the combination therapy is inadequate to eliminate all the tumor cells and something can stay back. Conclusions and outlook. Over the past few years, modern cancer treatments have impacted saving lives in tremendous ways. Treatments have been covered in the papers such as oncolytic viral therapy, proton beam therapy, have been less invasive and more precise in targeting the tumor, even if it has metastasized to other parts of the body, especially in pediatric patients. Precision medicine also makes up a huge part of major innovating cancer treatments because doctors and scientists have learned from their mistakes and went back to look at what was affected negatively, and hence focused on specific areas to improvise on it. So we can maximize the life of cancer patients. Combinations of highly targeted agents can be put together in a logical manner based on detailed genetic manner. Treatments such as oncolytic viral therapy and CAR T cell therapy are highly specific and engineered. What will the future hold? I think the future will seem a lot like this when you walk into a doctor's office. The patient will walk in with a memory stick that will have their electronic medical information and will have genetic information about their tumor and about themselves and their DNA makeup that will allow them to more rationally develop a treatment plan than we do now. So obviously creating extraordinary possibilities in terms of access and potentially quick diagnosis and targeted diagnosis and treatment, but also privacy issues. But what happens if you lose? This can be a problem. And so that's why it'll be important on the informatics side to figure out ways to encrypt this to be a password protected so that you know an individual's privacy can be protected. Cancer care in the next decade will be more personalized with more personalized tests and medicines available. Patients will be genetically profiled and genetic mutations will be identified and treated accordingly. Whereas we do need further research and development to effectively treat cancer, we have come a long way in terms of integrating ideas and innovation to save lives. Thank you.
Thank you.
I think you're on mute.
Can you hear me now?
Yeah.
Oh, okay. Does anyone have a question, especially from attendees? Or you can type it in the chat box or Q&A box. Otherwise, I have one question for all of you and anyone can answer. What was the most interesting thing you discovered in your research? Most interesting, most exciting thing? Yes, Taban.
This was, I guess, not necessarily exciting, but I think jarring. I discovered that metformin, which is one of the most commonly used treatments for PCOS, it's not only not FDA approved for treating PCOS, it's FDA approved for treating diabetes. So on one hand, it proves that diabetes and PCOS really have this like very close link of insulin resistance. At the same time, it is really concerning that like millions of PCOS patients around the world that are using metformin are using a drug that is not even regulated by like the Food and Drug Administration for treating that, which I think is really exemplary of like the leaps and bounds that we need to take in order to properly treat PCOS in the future.
Is there any side effects, metformin?
Metformin, there are side effects associated with hypertension, which can be concerning for patients, especially if they have obesity and are taking medication for their hypertension, because it can mess with their blood pressure as well. And as far as I know, that's one of the main symptoms. But I think another way we can look at it is like different phenotypes of PCOS have different symptoms and very different systemic responses to certain treatments. And I think metformin is more effective in some patients than others, depending on their phenotype. But the overall application of metformin as kind of this like blanket treatment really isn't the most effective way of going about it, which can, I think, result in a lot of inefficiencies within the medical processes and maybe even side effects. But yeah.
Wow, that's interesting. I should learn more about it.
Yeah, it's very prevalent.
Thank you so much. You did amazing job, all of you. Congrats and big applause. And have a nice weekend and stay safe.
Thank you so much.
Bye-bye.
Thank you.
Take care. Good job.
Oh, just a quick question. Sorry.
Yes.
I was wondering, is there an award ceremony of some sort, or will awards be announced towards the end of
I think they will send you an email about it.
I see. Okay, thank you.
Yeah.
Awesome.
Don't forget to check out your mailbox.
Yeah, I had a question too. Like, will we get like a recording of our presentation or something?
I think we will send those recordings to our moderators or panelists or who will grade them. Then they will send you.
Okay.
Okay, sure. Thank you so much.
No worries.
Thank you. Have a wonderful day.
Bye-bye.
Bye.